Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear students, welcome back in Pakistan International School TILES Virtual Learning System. This is session 2020 and 21, and this class is biology for students of grade 8, 9th junior. Dear students, today we are talking about some more very important topics regarding chapter number 5, cell cycle. And we have studied before about interphase stage, mitosis, significance and errors in mitosis. Then we have seen what's changing is going on in meiosis, what type of cell division it is, and now we will see its significance, like significance of meiosis, comparison between mitosis and meiosis, and we will see last topic of this chapter, that is apoptosis and necrosis. So let's get started. Yes, significance of meiosis, like what's importance of meiosis. We have seen the importance of mitosis as well, like that is the cell division which helps in, not only helps, but it is mandatory for the growth and development, cell replacement, regeneration processes. Meiosis is also having very significant role, and that role is for a reproduction and inheritance. Reproduction means production of new offsprings, new organisms. And inheritance means, you know, that the transference of characters from parents to the offspring. These two very important characteristics or significance or importance the meiosis division is having. So this reproduction and inheritance with meiosis, that was described in 1890 by German biologist August Weismann. What he saw, he pointed out that meiosis was necessary not only to maintain the number of chromosomes in the next generation, but to produce variations in the next generation. In meiosis topic, we have discussed in detail how the genetic variations occurs in the next generation. So when one cell, you can see here in the diagram, one cell is divided into 4 cells. So here you can see in meiosis 1, genetic variations is done. You can see this chromosome is having blue color and this is having green color. So some part is blue in the green and some part green in the blue. It means both of these have exchanged the genetic material. By exchanging the genetic material, by the process crossing over, variations in the next generation is done. These 4 cells are haploid. So these haploid, 4 haploid cells means half the number of chromosomes as compared to the parent cell, helps to maintain the chromosome number in the next generation as well as to keep the genetic variation in the next generation. How this all is done, we have talked about in the previous lecture in meiosis. But meiosis is essential for sexual reproduction. Mitosis is essential for asexual reproduction. And meiosis is essential for sexual reproduction. So the first very important significant role of meiosis is maintenance of the chromosome number in the next generation. How does it happen? If we talk about in humans, we know that diploid gametes are the germline cells, the sex cells like male cells and female germline cells. Like male germline cells are sperms which are produced in testes, and female germline cells are called egg cells which are produced in the ovaries of the ovaries in the female body. So the mother cells or germ or both of them, male and female cells, The mother cells are called the parent cell, which are the old one. They undergo meiosis. The meiosis step we have seen before in the previous lecture. To produce haploid gametes, male and female. From one cell, they produce 4 cells which are having half number of chromosomes. They unite to form diploid zygote. Now, what will happen here? You can see here the female cell and here the male cell. In the last, they produce 4 cells. Okay, remember one thing, for female sign, we put circle and under that plus. And for male sign, we put circle and over that arrow. These are the signs for male and female in biology. Most of the books you will find these signs, so don't be confused. So these 4 cells, these are mother cells, like from the female body, and they are the sperms, these are the eggs. Remember one thing, sperms are produced in millions of numbers and female egg is 1. So these 3 degenerate later on. So don't go it for detail because you will study this in the next higher classes. But from male, the sperm cells are in millions of numbers because after mitosis, they will get meiosis, then mitosis, and then meiosis again and again. So the haploid number of cells are produced. So what happened then? Female and male gametes, these are called gametes because that is egg and that is sperms. So when they unite to form diploid zygote, because now after meiosis they are haploid and that is also haploid, like that is having 23 number of chromosomes. So when this egg of 23 number of chromosomes will unite with any of this sperm which is having 23 number of chromosomes, all of 23 number of chromosomes, so it will come here 23, and for example this will come here, so 23, 23 and 23, that is 46. So that is haploid, that is also haploid. When they will combine, there will be 46 chromosomes. So in the next generation, diploid cell is formed while fertilizing egg with the sperm, which are having 23 number of chromosomes. So now the chromosome number in the next generation is maintained as 46. That's why meiosis is very, very important. Okay, so which undergo repeated mitosis and develops into diploid human being, like this zygote, which is having egg and sperms, both of the chromosomes, and there's a 46 chromosome. Now this zygote is diploid. Diploid, and then after fertilization forms zygote, and then there will be division. Now we have seen in the significance of mitosis, like development and growth will be done by mitotic division. So then mitotic division starts and a new individual is formed. I think, and I hope that you understood all this. That is, it seems to be very complicated, but it's so simple. But the complications and very detailed structure you will understand in the next higher classes. Many haploid fungi and protozoans. Haploid means it is having half number of chromosomes. Fungi, which fungi are having half number of chromosomes? Almost so many are there, like for example mushroom, haploid fungi, and protozoans. We have studied in chapter number 3, protozoans, animals like protists. So they, for example, We are having Euglena and amoeba. They are protozoans, and they are haploid. They produce haploid gametes through mitosis because they are already haploid. So they don't make more less. They don't make the number further less than haploid. So they go through mitosis through for doing this haploid chromosome number maintenance. Number three, we'll see which type of organisms are there. Plants. Plants life cycle show alternation of generation. Alternation means chain from one to the second and then second to one again of generation, because in all the plants there are two types of generations. What are these? We will see right now. The cells of diploid sporophyte. So two generations in the plant's life, that is sporophyte generation, and the other one is gametophyte generation. So sporophyte generation is the plant, green plant which we see around us. All the trees, all the herbs, shrubs and plants, they are sporophytes, the higher plants which are having xylem and phloem in them. And they are diploid in, their cells are diploid chromosome number. So they undergo meiosis to produce haploid spores. They produce haploid spores, undergo meiosis, which grow into haploid gametophyte. Then this plant undergo into several changes and they grow into haploid gametophyte generation. Gametophyte generation produces haploid gametes and through mitosis, because they are already haploid, so they maintain the numbers through mitosis. And when the gametes, because gametophyte generation forms gamete, that is called pollen grains and ovule in the ovary. So they produce gametes, ovary produce ovules, and stamen produce that is pollen grains, which are present, which are the parts of flower, to produce diploid zygotes. So when gametes, male and female gametes, they unite, so they form diploid zygotes. So zygote undergoes repeated mitotic divisions and they become haploid sporophyte. So how this all happens, we will see here. You can see this is a flowering plant, and for example this is hibiscus. So this is sporophyte, the plant which we can see, mature sporophyte plant with flowers. So in the flowers we are having gametophyte generation, germinated pollen grains, male gametophyte on stigma of the carp. Gametophyte generation, it means in the ovary, you can see this is ovary, it is having ovule in it, and that ovule is called egg. And in these anthers, these are the form pollens by, both of them are formed by meiosis. And what happened then, they form lots of mitosis and their number is increased. So the pollen grains, they fall on the stigma and the pollen grains just move into this tube that is called pollen tube, and they go to the ovule. This ovule is the gametophyte, female gametophyte. In that this egg is present, and the nucleus of the male gametophyte, that is called sperm, that which is present in the pollen grain, that unites with this egg, and this ovule becomes a seed later on. Some are having one seed, some are having more seeds. So you can see here the fertilization is done, and now embryo is formed. Embryo by mitotic division, like when we grow the seed in the ground, in the soil. So a small plantlet is, or seedling is going to be sprout out from the soil. So that is sporophyte, that is like made by lots of mitotic division. Now we are getting fruit. So ovule is present in that, and fruit is, for example, this is a plum or any type of we can say guava, any type of fruit, cherry, etc. So this is ovule. It is having this sporophyte plant, and this is the ovary becomes the fruit, and this ovule becomes the seed. Now this seed, when we grow in the ground, so after mitotic division, shoots and roots arise, and on the whole, a mature sporophyte plant we get. Here we can see here there is the blue one that is having this one blue that is having haploid number of chromosomes, this gametophyte generation, and here this gametophyte generation, and the other white one is all they are having diploid number of chromosomes by after fertilizing. So plant life cycle, if we see this in the cycle form, so this is fertilization, that is the mincing gamete, don't go to it. So zygote is formed, mitotic division and development is growing, embryo, this sporophyte plant, this whole plant is like formed, and then this plant under having some like parts like flowers, it is having some parts which undergo through meiosis, and it forms a gametophyte generation. uh, Meiosis, spores are produced. These are the spores which later on form pollen grains, and then mitosis is done, so again sperm is formed after mitosis is done, and then fertilization, and then again new organism is formed. This is called alternation of generation. It means from sporophyte plant, this one, we get gametophyte generation, and from gametophyte generation, again we get the sporophyte plant. So like that, gametophyte will change into sporophyte, and sporophyte will give rise to into gametophyte generation. This is called alternation of generation. The next significance of meiosis is production of variation in the next generation. Like we are having genetic variation, like all human beings are different, or kids are different from their parents. How this is happens? So we know that chromosome pairs in each parent undergo crossing over. So here the crossing over is going to be done, or you can see here the chromosomes are exchanged when they are this is called recombination or crossing over. Recombination means they recombine here. So after that, this is synapse when they make pair, and then they do crossing over. Here the genetic variation is done. So we all are different from our parents. So gametes fuse and form zygote, and its genetic makeup is different from both the parents. So meiosis also allows the species to bring variations in the next generation. So beneficial variations help organisms to adapt to the changes in the environment. If according to temperature, the organism cannot cope up the environment, so definitely they will die off. But with the variation, genetic variations, it can be easily adopted, like organism can adopt their environment easily. These were the very important two significances of meiosis. Now we will see comparison between mitosis and meiosis. This is interphase stage for both of them. So here mitosis is done, and in this side meiosis is done. So we can see here, first of all, the changes here. Here the homologous pair of chromosomes, like with 2 chromatids, but here synapse is done with 4 chromatids. That is prophase stage of mitosis and meiosis. Meiosis 1, because meiosis 2 is exactly same like mitosis. So all the changes are going on in meiosis 1, in prophase 1 especially. So here the homologous chromosomes do not form pairs, so no crossing over. So here the pairing of chromosomes is done, so the crossing over is done. Here the single chromosome, this is single chromosome, is attached or aligned at the metaphase plate, but here the chromosome pairs to form metaphase plate. Then in anaphase stage, chromosomes break an individual chromatid. This is one chromatid, this is other chromatid. They move on both of the sides of both of the poles to make two cells. But here the individual chromosomes are pulled downward, like a complete chromosome is now pulled on both of the sides. So daughter nuclei contain diploid number of chromosomes here, but here we are having haploid number of chromosomes, because they both are same and they both are same, so they get half here, but here all are different. So haploid number of chromosomes, each chromosome has two chromatids here, but each chromosome is having, here is one single chromatid, not complete chromosome. So we can see that some more points you can add, that in mitosis, there after mitosis we'll get two identical cells, but after complete meiosis 1 and 2 we'll get four number of cells, and here two number of cells. After mitosis we'll get diploid number of chromosomes in both of the cells, but after meiosis 1 we will get, or after meiosis we'll get haploid number of chromosomes. So here you can see diploid number of chromosomes. Number 3 point you can add here, that mitosis is done in all somatic cells of the body and meiosis is done in germ-line cells of the body, or in the gonads. Gonads means where the gametes are formed, like that is testes and ovaries in female. So like this we have seen like the comparison, that is so easy. And next one is mitosis is having only one set of phases, but meiosis is having two sets of phases, like meiosis 1 and meiosis 2. Now we'll see errors in meiosis. During errors in meiosis occurs especially in anaphase stage. Anaphase stage we have seen before. What happened? Chromosomes get separated. Complete set, a pair of chromosome gets separated in both of the like by shortening of the spindle fibers, like this complete and this complete. Okay, opposite poles. In anaphase 2, sister chromosomes are separate. So it is called disjunction. Disjunction, from the junction they are moving both of the sides, that's called disjunction. Sometimes the separation is not normal, and sometimes it happens they are not completely separated. Sometimes they get attached with one another, and sometimes like this set will not be separated and it will go on this side, and this is called non-disjunction. You can see here in this diagram in meiosis 1 when these are separated. So what happened with non-disjunction, one cell gets empty and the other cell is having all the set of chromosome here and the other cell. So this is called non-disjunction, when the cells are not separated easily. So in such abnormal gametes, when like they are gametes are formed here, you can see here in non-disjunction in two stages, like at meiosis 1 and here is meiosis 2. In meiosis 1, all the set of chromosomes get not separated and it just transfer to one side, and one is having not that set of chromosome. Here in meiosis 1, all is getting normally, but when there is meiosis 2, so this disjunction is quite normal. This is normal, but this is abnormal because this chromosome is not get separated, so it is just linked with one another and shifted towards one pole, and here no chromosome, so it means this is empty of that chromosome. And here when this cell divides, so the chromosomes are not present in both. So these are four gametes. They are this is, now you can see at the end you must get this type of chromosomes, but you are getting set of chromosomes here. When this will unite, with this one. So definitely in place of 1, we are having 3 chromosomes now, 1, 2, and 3. When this will unite with this one, we are also having 1. If this will unite with this one, we are having 2. So all the possibilities are abnormal now. Like any of the gamete can, male gamete or female gamete, that can be done in any of the two. So they can get the abnormal chromosome number. So in human beings, with the abnormal chromosome number, there can be lots of problems. Like if non-disjunction, so one cell will have 47 chromosome if one is non-disjunction is occur, and other will get 45 chromosome. Like both of the gametes are there. By fusion of this one, so we'll get 47 or 45. So normally we are having 46 number of chromosomes. So you can see here what can be the problem if there are 47 chromosomes. So there will be the problem that is called Down syndrome, and the baby which will be produced by birth, he will have this type of problem with having 47 chromosome. So that is called trisomy in other language. Like we are this is all the karyotype, like the gene chromosome structural and functional structural map which the scientists have discovered of human beings. Like these are same, these are same, these are same, all these are homologous chromosomes, and we are having, you can see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, and 21, 22, and this is 23. So we are having 23 set of chromosomes. It means when we get double, so we are having 46 chromosomes. But what happened at number 21 set of chromosome, pair of chromosome, here we are not having 2, but there are 3 chromosomes. It means at place of 46, the person is having 47 chromosomes. So now he will get Down syndrome, trisomy. Maybe you have seen in your life these type of persons. They are not normal actually by birth. They are having abnormalities in their like in their sexual life, in the and like all in their body structure and functions as well, mentally also. So if the person get where there is no chromosome in the gamete, so the person can get Turner syndrome, and that is called, where the chromosomes are 45, that is called Turner syndrome. So it means one chromosome is less. The person can look like this. This is a Turner syndrome. That's by birth problem. It is not treatable. It is not treatable, and with the medications and some physiotherapies and like that, like it can be we can make their life lifestyle change and life better, but that is actually untreatable because that is due to the chromosome and deficiency, or less chromosome in, or more chromosome in the cells than every cell. So there will be the problem that every cell then will be mitotically divided and a human being is formed. So that's why every cell is having the same type of pattern. Now we'll see, dear students, last topic of that, apoptosis and necrosis. Apoptosis and necrosis are two phenomena of cell death where the cell dies in the body. Apoptosis is the main programmed cell death. Programmed, like our body, our cell decides it to sequential and programmed to die. If what happened in that cell shrinks and become rounded due to the breakdown of cytoskeleton by enzyme. So during this apoptosis process, what happened? The cell shrinks and become rounded like this due to the breakdown of cytoskeleton. We have talked about cytoskeleton. That is a skeleton where microtubule and microfilaments are present in the cells to make the skeleton, so that it gets degenerated by the certain lysosomal enzymes. So it's chromatin. Now the chromatin material which is present in the nucleus, it undergoes condensation and nuclear envelope breaks. So in this way, nucleus spreads in the form of several discrete chromatin bodies. So you can see what happened. The chromosome cytoskeleton is broken down, and this, these are chromatin material just disintegrated, and that is like this is cytoskeleton is broken down. The nuclear material, all this disintegrates and condenses, and they form the separate nucleus spread in the form of discrete chromatin bodies. And then what happened? The cell membrane makes irregular buds, and those buds are called blebs. And these blebs break off in the cell from the cell. So those are called apoptotic bodies, which are then phagocytosed by other cells because they spread. So the white blood cells are everywhere, so they eat them and degenerate them. So this apoptosis can occur when a cell is damaged and undergoes stress conditions. So in an adult human, 50 to 70 billion cells die each day by apoptosis because that is programmed. Lots of cells are functionless, so they get apoptosis by this type of process. So apoptosis removes the damaged cells, preventing it from the getting further nutrients. Otherwise, they'll get nutrients and they proliferate in whole of the body. So that is the natural process. For example, during the formation of fingers in embryo, in the fetus, in the baby, so the cells between them undergo apoptosis and digits separate. You can see here, like when the fingers are like this, for example, so with them, in between them, before that there is a web with the skin. So this skin is removed naturally by apoptosis process before birth. So here we can see the natural process of apoptosis. This is the cell which is going to have this process of the programmed cell death. Then we are having necrosis, that is accidental death of the cells of the living tissues, and that is less sequential because that is done in any type of injury and the problem. So for example, infection and cancer cells, they are also gone through necrosis process. So they occur when that process occurs when a cell is given hypoxic environments, like if the cell cannot get proper oxygen, less oxygen, the cell started to die and accidentally die. During necrosis, there is a release of special enzymes from the lysosome. We know the lysosome releases the digestive enzymes, and digestive enzymes break cellular all the contents you can see here in the diagram, and may also be released outside of the cell to break surrounding cells also. And the cells that die by necrosis may also release harmful chemicals to damage other cells. So it bursts like this, so the nearby cells are for especially. That's why if the necrosis doesn't treat, so we can have the problem in surrounding tissue. So you can see all the cells lose their functional and they get swollen by absorbing the water, as well as and they break down by lysosomal enzyme by bursting. So very good example of that, spider bites. Spider doesn't bite, but there are some are very poisonous, so they bite, and necrosis is done in some area like that. By that chemical start to digest and break down the cells nearby its biting. Necrosis may be due to the lack of proper care. If we don't care the wound, so it can spread in whole of the leg, whole of the body. So this is a wound and nearby cells are going to be damaged. So especially this is happens in the diabetic patient and some chronic diseases like cancerous patients. So every wound has to be treated properly. So dear students, that's it for today. If you see this one exercise, what is cell cycle, what are its main phases? So that is about mitosis and interphase. S phase of interphase is important and cell can never divide without it. Why, how? We know in that chromatin material duplicates. That's why. So you have to write in detail. So, what is cell cycle? What are its main phases? So that is on first page of this chapter. What would you state the events of prophase of mitosis? How would you state? So prophase of mitosis, we have to write all of that. Make a list of events of mitosis. All the events of mitosis, you have to make the list and all what is going to be done in that, in the points you have to write, not in the detail. How is mitosis significance? So we have to write all the 3, 4 significance which is, we have studied. Describe the events that occur during the phases of meiosis 1. So meiosis 1, prophase 1, metaphase, anaphase, telophase 1, and cytokinesis. You have to write it. Describe the significance of meiosis. This is very simple. And contrast means comparison and differences. We have to write this. Describe necrosis and apoptosis, both of the paragraphs. Then question answers, like short questions. A nerve cell does not divide after its formation. In which phase of cycle? It is definitely, it is in G0 phase. Why it is done? You have to write it. How is cytokinesis different in plant cell as compared to animal cells? So we have to write about all the procedure of cytokinesis in both of them, which is done after mitosis, which is written in your book. What type of cell division occurs when our wounds are healed? So that is mitosis, which is done in regeneration. We have studied and you can write that. Plants do not make their gametes by meiosis because um, gametes are already haploid, like in the gametophyte generation, the spores which are produced, they're already haploid. So they undergo mitosis to keep and maintain the chromosomal number by mitosis. So you will write about this. So, dear students, that's it for today. I hope you understood it. This was your last lecture of biology. We have completed chapter number 5, which was very important. Just keep learning and remember me in your prayer. Wish you all the best. Allah Hafiz.